Okay, good morning for what is now the last of this series of lectures on DEB focusing on ecology and ecotoxicology, and this lecture is going to be devoted to applications to ecotoxicology with particular focus on problems relating to the environmental implications of nanomaterials, in large part because much of the motivation for giving these lectures derived through interactions with people in the UCCEIN. The lecture is going to, or the pair of lectures is going to be split into really three themes. I'm going to start with a short review of what we've already done, looking at just the basic concepts of DEB theory for individuals and populations. Then the great majority of the material spread over 2 sessions is going to be concerned with application of DEB theory to ecotoxicology. And then I'm going to end, since this is the last of the 4 lectures, with a few reflections on what we've done and a brief look forward. Probably the most important single thing I will say in this lecture on application to ecotoxicology is that I strongly recommend that anyone interested in this theme read the ebook by Chaling Yagar. It's a free ebook. It claims on the website it's a beta version, but it's more polished and more readable than many final versions. I've put the link up there. There is not only some very authoritative and readable text, there is a technical supplement that contains a lot of further additional information. So if you really want the take-home message from this lecture, it's read Chaling's book. So let me start with a brief review of what we've done in the first 3 weeks. In part to identify a few themes that will continue through this lecture, but also because I think there may be 1 or 2 people participating from CEIN who have not heard the first 3 lectures. Our focus has been Bas Kooijman's dynamic energy budget model, which is illustrated diagrammatically here for an animal. The basic idea is that food in the environment is assimilated, initially stored in a compartment called reserve, from which it's mobilized and used for growth, maintenance, development, and reproduction. And we've spent much of the last couple of weeks learning the rules, how you derive equations for this model, and some of its properties. The model has, in its full form, 6 state variables. So an organism is characterized at any given time by 6 numbers. The first 4 relate to the compartments you've seen in the previous model: the amount of reserve biomass, structural biomass, the total amount of energy or material that has been committed to reproduction, and a metric called maturity that relates to the state of development of the organism. In last week's lecture, we introduced 2 further concepts that relate to mortality, a concept called aging acceleration that is related to damage-inducing compounds, and a hazard rate, which is effectively just the instantaneous risk of mortality that is related to the total amount of what is called damage that has taken place within the organism. The model has 6 state variables, and depending exactly on what you want to do with it, you can come up with a different total for the number of parameters, but for many applications we can think of it as involving 12 parameters. One of the strengths of DEB theory that again I talked about in previous lectures is that some of these parameters are expected to have broadly invariant values across taxa, and others scale in a predictable way with size, and I'll make a few comments on that later on. But also for many applications where we're not trying to make predictions that involve all the state variables, we actually don't need all the parameters. In particular, there are a number of applications relating to individual organisms where we don't need to think about the parameters relating to aging, acceleration, and hazard. One of the model properties is that or the key model property, as I say, is that it relates the state of the individual organism to its environment, and the output, if you like, from the model is a series of plots of changes over time of a whole range of quantities: size, mass, elemental composition, reproductive output, and so on. And again, we've looked at quite a number of these. In last week's lecture, I then talked briefly about how DEB ideas could carry forward into population ecology, and that involved making a connection with standard population ecology theory. The starting point was that absent any changes in the environment, any feedback from the organism to the environment, the ultimate fate of a closed population is unbounded growth or extinction, and the long-term pattern of growth or decline is exponential. I wrote down an equation, the so-called Euler-Lotka equation, that can be solved in order to derive the long-run growth rate of a population in a constant environment. It involved two functions: the quantity I called beta, which is fecundity, the quantity I called survival. And using Bas-Kooijmans' DEB model for any given environment and a particular set of parameters, we can compute these quantities beta and S, and thus we can compute R. And later on in this lecture, we'll see an application of this to a system with a system involving nanomaterials. Real populations don't typically grow exponentially, and a population regulation involves feedback. And I demonstrated again in the last lecture that we could use individual-based models where the individual organism was obeying DEB rules in order to make predictions about population dynamics. I demonstrated that for an example of Daphnia populations, and later on in this lecture I'll demonstrate the same approach being used for a Daphnia population experiencing a contaminant. So we have a set of principles relating to population dynamics, and we've seen in previous weeks how these link tightly with a dynamic energy budget, or can be linked tightly with dynamic energy budget concepts. So why do we need theory, further theory, to address ecotoxicological questions, and in particular the effects of the environmental implications or ecological effects of nanomaterials? And I think there is a fairly well-defined and compelling rationale. The first and most obvious part is that contaminants in the environment have ecological impact at all levels of organization: individual organisms, populations, communities, ecosystems, and that's been a theme of the lectures up till now. A second and important one is that contaminants outside the laboratory act in concert with a whole load of other stresses. The same environmental insults that may introduce toxic substances into the environment may well be associated with a change in temperature, a change of pH. They may change the availability of food for an organism. We need theory that tells us how these quantities come together. And then there is a completely practical application, a practical issue, and that is that traditional approaches based on toxicity testing in no way can match the rate of introduction of new chemicals. Depending on where you look up, you come up with somewhat different numbers, but many places give you a number between 50 and 100,000 for the number of chemicals registered for commercial use in the US, and I got this guesstimate of 75,000 from a EPA website. Of these, probably less than 1,000 have undergone the complete suite of toxicity tests across a range of organisms, and this is in large part because of the overwhelming cost of tests. And Rinell, the CEN director, in a lecture, in several lectures, has quoted figures of 2 to 4 million as the cost for a complete suite of tests on a new substance. And obviously, even with doubling, trebling, 20-fold increases in budgets for testing, that's not going to happen and not going to provide the complete answer. So we need theory, and the reason it's in this course is that I certainly believe the dynamics of energy and elemental matter budgets are a component for this theory, and that Bass's theory is the obvious way forward at this point. These are general remarks. Nanotechnology has made the challenge tougher. If you want a reasonably precise, albeit arbitrary definition, nanoparticles are particles with a characteristic dimension of something less than about 100 nanometers, and that possess properties that are different from non-nanoparticles with the same chemical composition. And this means that not only are we dealing with many thousands potentially of different compounds, we're dealing with compounds of different size, shape, they're often manufactured with coatings and so on. There is no scope, there's very little scope for a full suite of testing on all these combinations. And in this slide I've given just a few examples: metal oxides, silver nanoparticles. We'll hear something a little bit later in this lecture about quantum dots, which are used in electronic applications. And when these get into the environment, there are a number of different types of potential impact. The obvious one is toxicity, especially since some of the applications involve nanoparticles that have been designed for their antibacterial or antifungal properties. That's obviously going to matter in the environment. And more subtly, and again related to DEB, there is the potential that nanoparticles entering the environment impact biogeochemical fluxes, impact nutrient cycling. So where do we gather information? Let me skip this slide. Where do we gather information information from in order to work to make studies of the effects of either toxic substances or, more specifically, nanomaterials? And there's a range of type of experimental information that is available. In recent years, it's been possible to get vast amounts of data on short-term studies in high-throughput robotic systems involving bacteria, cells extracted from many organisms, some embryonic studies, and a range of molecular studies. This slide, again taken from or modified from a slide of Dr. Nell's, suggests that these kind of data can be gathered at the level of hundreds of thousands of information points per day. As you move upscale in biological complexity and ecological relevance, you reach the point of tests on a large organism, such as a small mammal, or in the natural environment. And there, at great expense and with great difficulty, it is possible to do small numbers of experiments per year. In short, there's a trade-off. We can gather vast amounts of data at a level that is going to give information on suborganismal mechanism and very little information on its ecological significance, and we can get very small amounts of information at an ecological level. And one challenge that those of us that work in this field face is to somehow use this information from the molecular and cellular studies to guide, design, and interpret ecological experiments, given that ecological work is time-consuming, expensive, and needs to be well-focused. And again, the suggestion in this lecture is going to be that DEB models can help in this activity. I want to emphasize, before going on to the specifics of how we apply DEB models, the limitations of other approaches. And the sort of information that you get from standardized toxicity tests is often metrics such as the EC50 or the EC10, the level of a contaminant that gives you a 10 or a 5%, a 50 or a 10% a reduction in some measure of performance. And I illustrate this with a pair of pictures taken from a very nice paper out of Bas Kooijmans' group published a couple of years ago. These are pictures of growth on the left panel. Let me get a pointer. Growth on the left panel and reproduction on the right panel of C. elegans exposed to one particular contaminant. And now suppose you want to calculate an EC50. Well, from the left-hand panel you would ask, how much change in length do I get at some time? Or from the right-hand panel, how much change in reproduction? And when I look at the next slide, I've carried over again the growth panel as illustrative. If you estimated the EC10 or the EC50 or the EC10 or the EC5 for these for length and for reproduction, the continuous lines tell you what you would estimate for the EC10 if you work from length. The other continuous line, what you would get for the EC5. The three broken lines say what you would get from the reproduction panel in the previous line. And the take-home message is that the number you get is related to the duration of the exposure and to the metric and to the biological process length or reproduction that is being used. And since tests involve different organisms and different with different protocols, it's very hard to see what the ecological implication of any number in this particular picture is. So we need something that is more biology-based. And the key argument for using dynamic energy budget theory is that toxicity experiments and modeling involve a bunch of unavoidable choices, and that DEB theory can contribute in some way to mitigating the effect of these choices. For a start, you have to choose what organisms you're going to work with, and there is a suite of so-called model organisms that are used in many, many studies. DEB theory has recipes that we've touched on in the last few weeks for interspecies comparisons. That is, we can make predictions from one species from information on another. This suggests that we can move past model organisms. Next, and important, the DEB theory, the DEB model that I showed in the opening slide can be coupled to separate submodels that take account of something that we know, of some things we know about the contaminant. In particular, we can describe the processes of bioaccumulation within the organism, that is, how fast a contaminant enters the organism and where it goes once it's in the organism. And we can provide representations of toxic action. And in a growing body of literature, these submodels of bioaccumulation and toxic action have come to be called toxicokinetic and toxicodynamic models. I think these terms derive from earlier terms, pharmacokinetic and dynamic, but they mean the same. They mean the same thing as bioaccumulation and toxic action. Then I noted in the previous slide that the interpretation of toxic effects depended on the endpoint chosen. I showed a difference between using an endpoint related to growth and one to reproduction. The DEB model parameters relate to basic biology, not to arbitrary choice of endpoint or protocol, really a point I already made. I've already pointed out that we have a recipe for making individual to population connections. So if you have information like again in the previous slide related to individual organisms, you could say what a large number of these organisms experience a common environment do, for example, using DEB IBM. And although I'm not going to talk about it in any detail in this lecture, or this period of lectures, just briefly touch on it once, a DEB model can treat the response to multiple stressors, and I think in the bigger picture this is one of the most important strengths of the approach. So from an ecophysiological perspective, if you like, from the perspective of the whole organism, what do toxicants do? There's a range of things that they can do. They can reduce the rate at which an animal feeds. They can increase maintenance demands, for instance, through costs of detoxification, costs of various other repairs. They can lead to slower growth. They can lead to, as we saw on the previous slide, they can lead to reduced reproduction, and most obviously they can lead to increased mortality. Importantly, these effects are not independent of each other. For instance, a reduced feeding will typically lead to lower growth and or reduced reproduction, depending on the organism's priorities for energy allocation. So these effects can all be either direct effects or indirect effects, and we need a flexible framework within which to explore these possibilities. And the approach here is to go back to our basic model and identify what I've previously called fluxes, the flows of energy or elemental matter between the boxes and the branch points like this one here, and then to note that the potential effects of a contaminant either in the environment or within the organism can be to influence one or more of these fluxes. So for instance, it could influence this thing called JEA that represents the rate of assimilation. It could lead to an increase in somatic maintenance. An endocrine disruptor might well impact the parameter kappa that represents partitioning between growth and reproduction, or it might impact development and hence have an effect on maturity maintenance, and so on. You can envision a load of different ways in which the flows in this model could be impacted by contaminants. And the applications of DEB theory in ecotoxicology effectively involve asking how the parameters of the DEB model are impacted by the environment within the organism or outside, as represented in the various submodels that get added. I'm going to illustrate through this through the next part of this lecture how this process actually works by showing you a few examples of projects that are ongoing in the UC Center for Environmental Implications of Nanotechnology. And each of these has been chosen in order to illustrate a different way in which DEB models can be used. I'm going to show some results from experiments on marine phytoplankton. I'm going to show some results from a study on marine mussels where it was possible to make this individual to population projection that I talked about. And then I'm going to show a study on bacterial population dynamics, which lets us understand better the connection between cellular level and subcellular level toxicity mechanisms and cell proliferation, which is a population phenomenon. And after I've shown these examples towards the end of the second part of this morning, I'm going to show a non-nano example that illustrates the use of DEB IBM as a way of as again a way forward for toxicological problems. So let me start by saying just a little about the effects of some work illustrating the effects of nanoparticles on phytoplankton populations. And this work used Bas-Kooijman's standard model, but very quickly recognized that the model was far, far too complicated for the application in hand, and therefore took a simplified version of the model in which the focus was only on 2 I-state variables, and instead of a huge suite of parameters, there was only a need to consider 3 parameters, with 2 parameters characterizing the toxic effects. So by making a series of well-defined systematic assumptions, it was possible to simplify the model to something that was practical for a relatively simple study. And the sort of data that was obtained was very simple again. It was simply data on exponential growth of 2 species of phytoplankton exposed to two different contaminants. There's far more in the paper I cite at the bottom, but that's just 2 sample graphs. The DEB model for phytoplankton simplifies to a very small number of equations if you make a series of assumptions. One is what I call V1 morphe that I again talked a little bit about in a previous lecture. Essentially it assumes that all rates scale with the structural volume or the structural mass. Secondly, it was assumed that the food environment was constant, so the reserve to structure ratio came into equilibrium, and the end result is that it's possible to describe the population dynamics by the simplest equation that ever exists in population dynamics, the simple equation for exponential growth, where there is a growth rate little r that depends on a couple of parameters. One relates to the rate of reserve mobilization, and the other is the scary DEB parameter combination little g that you've seen in a few other situations. Oh, sorry, and there is also somatic maintenance. The assumption was made in this work that the effect of the stressor acts on both the rate parameters, Ke and Km, and what's more, it does so by through the same factor. That is, Ke, the rate of reserve mobilization, is reduced by the same amount, by the same proportion that the parameter Km, the somatic maintenance rate, is increased. If you assume there is some no-effect concentration, the quantity we've called CNEC here, and there is a parameter that here, a K with subscript E, with subscript C that characterizes the strength of the interaction, then you have the possibility of fitting these parameters from using the data from these very, very simple graphs. And when you these are the 2 parameters. And when you do that, you come up with a an NEC with this particular value. That's a very piece of basic analysis of the sort that is done, that has been done using DEB models for at least 15 years, since some early work involving an approach called DEB-tox was published. But what is interesting about this is that it's possible to now compare what came out of the DEB model fitting for the no effect concentration with a series of experiments performed on subcellular processes. And the details of these don't matter, but they're again commonly used in high content screening studies. This is a mitochondrial membrane potential, a measure of membrane permeability, and in particular a measure of the level of reactive oxygen species, which I've talked about, which I talked about in last week's lecture. And what we see is that the crude estimate, which has error bounds along the range I'm showing with the arrows here, the crude estimate that we came up with from the DEB model fitting appears to be kicking in at least something like the same level as the quantities, as these various subcellular quantities obtained from high content screening. So if you're somebody who wants to advocate the use of these subcellular processes to characterize toxicity, this would appear to be very good news. This is saying that the information that's obtained from a series of essentially snapshots is pretty much consistent with what we get by fitting the dynamics over the much longer scale. But it's not always that simple. And here is just an example of another set of studies of phytoplankton growth, this time by Louise Stevenson, a PhD student at UCSD. And if we imagine that the early stages of phytoplankton growth look like what's been shown here in essentially exponential growth of one sort or another, over time, as you well know, population will grow and eventually saturate out at some carrying capacity. And the right-hand plot here shows the toxicity of this time freshwater algal cells exposed to silver nanoparticles during the phase of exponential growth and later on. And the basic message is that the effect later on is much, much less than is observed during exponential growth. And what's more, there are changes in the environment. The particles aggregate more when added to older cultures than to newer cultures. In other words, there is the possibility of some feedback from the environment that interferes with physical-chemical processes, and this is very much still work in progress. But again, it demonstrates that even in the simplest context, which is the phytoplankton example, we need to be working within a framework that will let us take account of feedbacks if we intend to carry insight over to real ecological contexts. The second example that I want to say just something about is the effect of metal oxide nanoparticles on populations of marine mussels, because that illustrates a different way in which the theory can be applied. This work again used Bas-Kooijman's standard model. Again, it used a simplified version of it. At least to date, there has been no consideration of either of the variables associated with mortality, but otherwise pretty much the standard model was used. Not all the model parameters in this case could be measured from the studies available, but there's been previous work fitting DEB models to mussels, and as I pointed out before, there's possibility of using related information. So there was actually the potential, due to the special features of DEB, to use existing literature information. It still left toxicity parameters to be estimated, of which 2 turned out to be unimportant. And here is an example of the here's an illustration of the approach, and it involved experiments largely done by Shannon Hanna, who recently completed his PhD here at UCSB, and modeling done primarily by Eric Muller. There were a series of basic measurements on individuals performed at different food levels. I've illustrated a few of them here, and they relate to exactly the quantities you would expect based on what I've previously talked about. Essentially measures of size and the relative proportions of somatic versus reproductive tissue in the organism, something about the bioaccumulation of the contaminant, something about feeding rate, and something about oxygen consumption rates, respiration, which I've talked about. The key thing is that all of these quantities are related to the DEB model prediction. These could be used to estimate toxicity parameters in a representation not at all unlike the one that was used in the previous work on phytoplankton. So now it is possible, with assumptions about survival, to figure out what would be the lifetime reproduction. And that is a formula that I wrote down at the start of the lecture. And if you prefer, you can go with little r, which is just a different metric with the same ecological relevance. And we get the reproductive output part from the DEB model. We get the survival from assumptions relating to the known life cycle of mussels. And this slide illustrates the kind of data that could be obtained. You're able to see how the change in length over 12 days, over the duration of the experiment, varies with exposure. You can see how accumulation varies with exposure, and you can see here how survival varies with exposure. And note that except at the very highest level, the effects on survival were, there was no impact on survival. Consequently, what you see is that sublethal effects, not surprisingly, kick in at much lower levels than mortality effects, again justifying the focus on these effects. And Eric Muller computed the following graph that illustrates the application of the method, and this is the one example I mentioned where there would be two simultaneous effects looked at. You can think of the line that is appearing down this graph as effectively giving us an indication of where the EC50 in lifetime reproduction would occur. The value at which it occurs depends on the amount of food environment. It depends on the amount of food in the environment. But the really important effect, the really important outcome, is that the EC50 for the lifetime reproduction is actually much, much lower than the EC50 that you get for the individual physiological rates. In other words, this is again a situation where short-term experiments and the projection from a DEB model will point you in different directions. And in this case, it's saying that you would be overly optimistic if you based your hazard assessment on purely the short-term physiological experiments. So I want to take the break now. It's almost exactly 9:40. When we resume in 10 minutes, I'm going to illustrate another example, again from CEIN work, where there is a much larger effect played by feedback, and where we have to introduce some deviations from the standard DEB. I'll then go on and talk a little bit about an application of DEB IBM, and after that wind things up. So the plan is to resume in 10 minutes from now. Thank you. So I'm going to resume for this second part, and the next thing I want to do is talk through a somewhat more detailed application of DEB concepts to again a nano-related study. And unfortunately, this slide starts with a typo. It should of course be Pseudomonas. But the example that I'm going to talk about relates to a series of experiments done by John Priester, Trish Holden, and a number of collaborators at UCSB on the effects on bacterial growth of exposure to quantum dots consisting of a matrix of cadmium and selenium with some coating around them. And the sort of thing that goes on here involves both what I would call DEB processes within the DEB processes as characterized by the standard model representing reserve structure, aging acceleration, and so on within the organism, plus a series of transformations that involve the quantum dots and their dissolution. So if you start with quantum dots here, they can be dissolved and can produce a dissolved cadmium. There can be exposure to the dissolved cadmium, and that can produce and that can have toxic effects. And as in the standard DEB theory, that impacts the damage-inducing compounds. There can also be effects on growth, so there are effects on growth predicted here, and that's just one path, because the quantum dots themselves may have an effect, and so you have to consider exposure to the quantum dots. You have to consider whether the quantum dots are themselves toxic, and if so, what is the effect of the contaminant. And the entire collection of processes described here impact the observed population growth that you see. And the strategy that is used, the strategy that was used by Priester and Holden and collaborators, that is quite common in this type of study, was to contrast the response of the populations to quantum dots to the effects on population growth of exposure to the same amount of dissolved cadmium. The assumption is that cadmium is the toxic material here, and so the idea is to look first at the effects of cadmium alone. And what that means is that in terms of the processes described by the box in the previous slide, I'm ignoring everything along the bottom line here. And this is work that was led at UCSB by Tim Flanschek, and the figure at the bottom right shows a series of bacterial growth curves, that's population against time, for Pseudomonas exposed to steadily increasing amounts of dissolved cadmium. And the first thing we did was to focus on just the control, the top curve here. And to our considerable frustration, we found that we could not fit the control to the simplest DEB model that where we pulled out the box. And there were a couple of additional variables that were needed. One was no surprise. There was a measure of acclimation that, if you like, relates to the lag that is often observed in bacterial studies at the start of the experiment. But even after taking account of that, there was a basic asymmetry that you can actually see in this curve. The exponential growth starts very rapidly, and then there is a very, very slow approach to the maximum. And we finished up having to be empirical in the way we characterized that by inventing a variable that characterized the environmental degradation that was treated as a DEB product in the DEB sense. But once that was done, we did have the controls fitted. And then we used a Basque-Lyman standard model with the two extra variables that I'm showing here, acclimation energy and this measure of environmental degradation. And that gave us a model with 6 DEB parameters, and then a variable number of other parameters depending on how many DEB processes we thought were potentially impacted by the cadmium. And what we were able to do was simultaneously fit these growth curves with different models, plus one other set of data. We were able to, we made an assumption, which is actually somewhat different from what is done in the DEB3 book, that we could identify the level of reactive oxygen species, ROS, something that was measured, with the damage-inducing compounds, or the aging acceleration variable in DEB. And what that meant was that since we knew the time course of the concentration of ROS, we not only were fitting models to these data, but to one piece of supplementary information that was coming out of the DEB model. And then, having done that, we were able to conclude that a model that assumed the primary toxic effect occurred on the rate at which the bacteria absorb resource from the environment, and a direct effect on mortality best fitted the data. This was all still working with cadmium, with dissolved cadmium. The next stage, which again I think is a strategy that is going to be followed in quite a lot of work on nanomaterials, was to go back to the schematic I showed originally, where we had both effects from dissolved cadmium and effects that were directly associated with the nanoparticles, the quantum dots. But now, having come up with a model of cadmium toxicity, we imposed a new rule on ourselves, which said that the cadmium toxicity model, including its parameters, was set in stone. We had to work with that model and with its parameters, and then we could figure out the implications of the various ways in which the quantum dots could have an additional effect and relate these to the data. And as you see in this picture, there's a lot of different ways in which quantum dots can change things. This particle here, for instance, might dissolve, and that introduces cadmium ions into the environment. The quantum dot might associate itself with a cell membrane and produce effects on the cell through being associated with the membrane. It's possible that the nanoparticle actually enters the cell itself, acting as a kind of Trojan horse that gets a large dollop, if you like, of cadmium into the cell, in which it can have its effect. There can be ROS produced due to this interaction here of the quantum dot of the cell membrane, or there can be ROS produced inside the cell. And again, there is a large range of possible mechanisms. When you operationalize this into a model, you finish up with a very large number of possibilities. Even after we eliminated some, we finished up with, I think it was 16 that had to be 16 possibilities that had to be compared. We had some limited information on quantum dot dissolution in the absence of bacteria, and that was fairly critical for what followed. So we knew something about these rates. Again, the information on an auxiliary quantity beyond growth and reproduction turned out to be critical, and again it was at the level of ROS, the reactive oxygen species. This work actually stalled for a while because when we attempted to use standard model selection methods on the range of models that I alluded to a minute ago, a good number of them fitted the data. I think we could characterize them as the good, the bad, and the ugly, and we could eliminate the ugly, but there really wasn't a lot of difference between a bunch of others. But then the focus on reactive oxygen species did allow some real model selection, and the slide at the bottom right here illustrates how that works. We invented a metric which involved a comparison of the observed level of ROS in the experiments with cadmium and with quantum dots, and the corresponding quantity with a cadmium, and we took the log of it. The details of the metric don't matter. The key thing is that it involved a comparison of the new data with the data that we'd used for the previous model, which was fixed. The slide here illustrates the three best models. We had 4 data points for ROS near the end of the study, 1, 2, 3, 4 shown here, and the message is that the best model, broadly speaking, followed the pattern. The next 2 models were well off in terms of ROS, though pretty well indistinguishable in terms of residual sum of squares effect on the growth curves, and that let us identify some specific mechanisms. And that, I think, leaves 2 take-home messages. One, which I've come at from different directions, including several discussions of respiration so far, is the value of the fact that DEB gives you predictions of other quantities than just growth and reproduction and so on. But it also illustrates a challenge. The inferences we made here are only valid if we were legitimately identifying the ROS with one of the DEB state variables, and there is a whole range of challenging questions as you get more biochemically specific information available on something other than just the flow of the major elements CN, O, and so on, and how you relate these to combinations of DEB variables. And I think this example illustrates both the power and the dangers of the approach. I now want to end the overview of different ways in which you use DEB in Ecotox with a non-ecotoxicological example. I mentioned in the last lecture that, in fact, I illustrated some data from the application of DEB IBM to modeling the population dynamics of Daphnia. You may recall that in order to get a population data to come into congruence with model predictions, all we required was the standard DEB plus a separately parameterized representation of food-dependent or energy density-dependent mortality for young individuals. Ben Martin used this model in a study of a relatively simple example of a contaminant effect, where again there was data on individuals and on populations in a chemostat. So let me remind you just remind you of the DEB scheme, which is shown here, and the graph on the right shows the effect of schematically the effect of a control and of one contaminant, but there's actually a lot more data, and it's taken from this paper by Thomas Price, cited at the bottom of the slide here. The black curve is the control, the turquoise curve is what happens in the presence of the contaminant, and broadly speaking, there is no effect on growth of this particular stressor. It goes in there and growth carries on at much the same rate as before, but there is an effect on reproduction. This is cumulative reproduction against time, and there is substantially less reproduction. And that alone reduces greatly the number of possible modes of action, the possible ways in which the possible DEB parameters, if you like, a stressor could be impacting. And I simply want to illustrate the effects of one. The model used was essentially the same model that I talked about last time, with the same parameters, but with the toxic effect parameterized from data like this. That slide, for some reason, came up twice. The effect on reproduction is illustrated here, and again, that's just saying where the parameterization came from. And the next sequence of slides shows what was predicted for the chemostat experiments. The fact that the control fits is no big deal, because the fit and the control is essentially the same information that we were looking at in the last lecture in the study of the population. So this was the tuned information. But now here are graphs from two separate concentrations of the toxicant, and again the agreement of a model and data is fairly striking, and this is the DEB model out the box except for this additional mortality and one assumed mode of action, albeit for a particularly simple case, because it's right at the end point. It's at the level of conversion of reproductive material into eggs that the stress is happening, or conceivably on embryonic survival. It's at that level. But again, it illustrates that it is possible to work. It's possible to take a DEB model, put it into this now a relatively well-defined and usable piece of software, and come up with predictions at population level of these populations in the lab, and get something that is something that is consistent. So pulling all this together, I think there are a series of messages that relate to ecotoxicology. The last example, actually the last example and the previous example on the quantum dots between them illustrate that IBM's structured population models can relate information on the individual, and in the case of the quantum dots, the suborganismal, through to population dynamics. And this is expressing the philosophy that I tried to talk about in that trade-off slide at the start, where you want to relate the information that is cheap and fast with the information that is slow, expensive to obtain, yet important. The underlying message of everything is that this abstract representation has a lot of practical value, because I've taken examples from a whole range of different contexts and taken the same representation. A reserve means a very different thing in a bacterial cell from in a mussel, and yet the same representation seemed to be pretty effective. The quantum dot example illustrated the importance of feedbacks from the single organismal processes through to the environment. In that particular case, we didn't understand what the feedback was, and we had to put it in a somewhat arbitrary phenomenological way. Environmental degradation might represent something like oxygen depletion in the cultures, but it could represent a whole collection of other phenomena. The freshwater phytoplankton work of Louise Stevenson may be giving us more mechanistic insight on some feedbacks, but that work is still in progress. And then the marine mussel example let us see that ecological effects, as characterized by in that case by the long-run growth rate, could kick in at different levels. In the case I looked at, it was a lower level than would be predicted from suborganismal studies, but it could go the other way with different processes impacted. And I at least don't see a way in which we would have got at this without using either the DEB models or something like them. Beyond this, for ecotoxicology, I reiterate my opening remarks, which is to get more specific information and methodology. Download Chani Yagra's book and follow it through. It is full of fascinating detail. I want to actually use the last few minutes of this last lecture in the series to just make a few broad comments about what we've done and to look forward. I don't intend to give a recap of all the lectures. I'll be posting the remaining slides, and the slides themselves act as a recap for the lectures. But I want to pick first on a couple of distinctive features of the model, and to move on from these to make perhaps one comment about the future. And the first question relates really to the abstraction of the modeling approach. And one way of thinking about it, a phrase I took away from one of the earlier DEB symposia, is that you can actually think of a DEB model as a sophisticated regression model that relates a series of output variables, the ones I identified at the start, growth, reproduction, respiration rate, and so on, to a series of input variables, which are the environmental variables, food availability, whatever other stressors are present, and so on. And viewed from that perspective, it's a state space model, and state space models frequently have invisible variables as intermediates between the inputs and the outputs, and reserve structure, and well, these two in particular, but also some of the other variables would be simply, from that perspective, statistical intermediate variables that work, if you like, that provide the success that's described because we've got the appropriate dimensionality of the problem. That there's a lot of things that can be characterized with the number of state variables that we've got, and I strongly suspect that that is at least part of the story. DEB models work because they represent a remarkably powerful regression tool. But they are based on a perspective of how an organism works. They differ from purely statistical state space models in the sense that you're not just taking any old function that relates the inputs to the reserve and another any old function that relates the reserve to the various components afterwards. It's not just a case of functions that you could fit with polynomials or splines or whatever you want. There is a biological story that underpins these. And for me, that represents why we need to keep probing the relationship between these more abstract variables and the various observable quantities. They're important. Their interpretation is really important if we believe that the DEB model represents some fundamental design principles that are present in that are involved in DEB, and not just a strong regression. Because if you think that the model represents some strong design principles, then you can actually make some strong statements about evolution using, and there's a lot of scope for development of new evolutionary theory using using DEB theory. And there is a very impressive start to this has been made in a paper that came out at least 5 years ago by Bas Kooijman and Tineke Troost, listed here. It describes the early evolution of metabolic organization. It's sufficiently far from my area of expertise that I can't evaluate the details, the details of that. But if this is a productive way of thinking, then DEB has to be more than just a regression model. It's in some ways an abstract representation of design, and I'm pretty optimistic about this, the potential of this latter approach. The second comment I want to make is really to simply mention what is probably the strongest and most distinctive feature of DEB that I have not included in these lectures, and certainly will be included in for people who follow Bas's telecourse and or go to the hands-on course in TESOL. And that is that one of the strengths of the DEB model is that the same model can be used to characterize all the life cycle stages of an organism, and not just the juvenile and adult stages, which we've already talked about. And I want to illustrate this with a set of slides that I've adapted from slides given to me by Laura Peccary, which follows the life cycle of a fish. Suppose you start with an embryo. What does an embryo do? An embryo, largely speaking, consists of a large amount of reserve material and a single cell. And the development of the embryo, the progress through the various developmental stages, involves utilization of energy from the reserve in order to build structure and to add complexity to the organism. So embryo starts with a load of reserve and an absolutely tiny amount of structure, and let's arbitrarily say no maturity, and then over time reserve gets utilized, structure appears, reserve is depleted, and the process of development leads to an increase in maturity. Eventually, the reserve is getting depleted, and this mode of development is no longer possible, and there is a transition to a feeding stage. And actually, what's showing there is actually a schematic of a small young fish, a hatchling. This individual starts feeding. It gets energy from food, goes into the reserve, and it partitions the mobilized energy from the reserve in very much the way we've been talking about in the course. The kappa rule is applied, and you get a steady increase in maturity. This amount of maturity steadily builds up, and then you reach the point where you have a much larger individual. The maturity variable is increasing, still has a bit to go. Everything is proceeding with no changes in allocation until the fish becomes an adult. And now it is assumed that the process of maturation is ending, and the adult in many cases is assumed to be still feeding, though that's not absolutely essential for the formalism. A reserve is being utilized, but the branch that went to increasing maturity is now going to reproduction, and eventually reproductive material builds up and is spawned. The fact that we had a model with 6 state variables and 12 parameters, when we were talking about growth and reproduction, sounds like we have a pretty complicated model. The fact that we're now saying we have a model with 6 state variables and about a dozen parameters that can characterize all stages of a life cycle suddenly says we have a rather parameter sparse model. It is in many ways remarkable that you can capture all these different stages in a life cycle with a relatively small number of state variables and parameters, and it's actually only 4 if you take account if you don't want to involve consideration of mortality. I talk that through because I think that probably overall there are 2 really distinctive, strong features of DEB that separate it from other approaches to bioenergetic modeling. One is this full life cycle aspect. The other, which I think I've already said quite a bit about in the course and demonstrated through the Ecotox example, is the interspecific body size scaling relationships. I want to end by asking what's next. I've presented what seems to be settling into being a fairly coherent, increasingly applied body of theory that explains or is consistent with a lot of observed phenomena and has the various strengths that I've described. But theories in biology are not static quantities. Theories in biology evolve, and they can evolve slowly or they can evolve fast. And there is a long history of theory in ecology and of evolutionary theory that illustrate the processes. It's a mixture of really the classic evolutionary processes. Theories mutate, people change features and decide they're slightly better with this feature or that. They drift, which probably relates to fashion among among scientists rather than any strong rational mechanism, but they also experience selective pressures. New discoveries, empirical information causes theories to make small or large changes. And I am very reluctant to believe that the current implementation of DEB is where all this is going to end. It's very successful, which means it could be an analog of one of these organisms that has lasted through huge amounts of evolutionary time just because it's supremely well adapted to a range of problems. But I'm disinclined to believe that. I'm far more inclined to believe that, in particular, as we get increasing understanding of homeostasis, regulatory mechanisms from systems biology, some of the ideas that go into the DEB model are going to be replaced with the current DEB model are going to be replaced with others, while some of the core concepts, like the importance of obviously the importance of energy and mass flows, but also some of the subtleties of homeostasis are going to survive. I could be completely wrong there. I don't know how it's going to play out, but the message with which I really want to end this set of lectures is that if you don't believe that what we've been talking about is the last word, jump on it, and I'm waiting for somebody smarter than me to develop DEB 2.0, or if you prefer the language, next-gen DEB. And thank you for listening over what amounts to 4 weeks. Thank you.